Hi, my name is Julia Schernicki, and I would like to talk to you about glial cells and nervous system mapping. The National Academy provided one of the most important congresses of my life. For those 3 days, I left the armory feeling refreshed and ready to face the world. Even once I left the conference, I brought that pride back with me. Of those 3 significant days, the amygdala speech given by Dr. Taylor sparked a curiosity within me. A curiosity that was always inside of me, and one that will never leave. I began thinking about the brain and its circuits that it had within. The interest overwhelmed me, and I kept researching until I stumbled upon glial cells. These cells were once insignificant. They were thought of as the garbage truck of the central nervous system. But recently, these cells have been discovered to have a great effect on our nervous system. During the Congress, Dr. Jill Bolt-Taylor talked to us about the amygdala, a section of the brain that determines whether or not our body feels safe. Since her talks on TED, Dr. Taylor has been infamous, and it didn't go any differently in the Congress either. The scholars at the conference, my generation, admired her bravery to talk about her stroke of insight and willingly absorbed every single word that she said. As Dr. Taylor continued to talk about her stroke, I became interested in neurotransmission and what would happen if something went wrong. That night, I reached a personal epiphany. I realized that the secluded highway of neurons, known as the central nervous system, can be affected and altered by different types of cells. I knew that in order to share my realization with the world, I needed 2 things. 1, a cell that would prove me right. In my case, they were glial cells. These cells were known as the garbage truck of the central nervous system. But then, after the 1980s, these cells became very well known for their effect on the neurotransmission. And 2, a clear understanding of neuropathic associations and signaling pathways. Ironically, what better way to understand signaling pathways than a clear 3D representation of a brain? Unfortunately, in the day and age that we live in, there is no cure for neuropathic pain. Chronic neuropathic pain is seen as a continuation of electrochemical signals fired from neurons. Nerve injuries or lesions are often the most common source of neuropathic pain. Before neuropathic pain develops, a nerve lesion attracts glial cells to the site. This attraction makes some scientists speculate that the glial cells are the cause of neuropathic pain. Sadly, there is no way to fix these defective neurons, and those with neuropathic pain can only deal with their pain through various amounts of medication. Chronic and neuropathic pain are considered neurodegenerative diseases and are often forgotten in the medical research field. As a starting point, a clear understanding of neuropathic associations and signaling pathways is imperative to understand neuropathic and chronic pain. Pain models are often used to understand the associations of neurons and other cells, such as glial cells, in neurotransmission. The purpose of my proposed study is to solidify the glial effects on calcium and glutamate-dependent neurotransmissions. This further proves that the once isolated central nervous system is now a series of neurons that can be affected by various other cells, such as glial cells. There are 5 different major types of glia: astrocyte or astroglia, microglia, oligodendral glia, and satellite cells and Schwann cells. Although these cells are categorized under a general glia cell, they are very similar in function. However, astrocytes or astroglia have been known to affect neurotransmission more often. It is important to understand that glial cells do not carry any action potentials when it comes to neurotransmission. Soon you will see a video done by National Neuroscience. In this video, scientists make the brain transparent by removing a lipid layer and putting the brain through a light microscope. This makes the brain clear. A whole brain can label different molecules, such as astrocytes, which in this case are specifically blue.
We're heading into the center of a mouse's brain, into the hippocampus, where memories are formed. Looking up, you can see neurons projecting to the surface of the brain. This is the work of Carl Diceroth and his team at Stanford University. By making the entire brain transparent, they were able to image it using a light microscope. They call the new technique clarity. Existing techniques for studying the brain's wiring are often limited to looking at very small volumes of brain, or they don't allow you to label genes or chemicals of interest. The advantage of clarity is that you can label lots of molecules in whole brains. So how do you make a brain transparent? The thing that obscures the view is fat. Lipid layers surround each cell. To remove them without disrupting the cell structure, the team used a hydrogel to create a mesh to hold the rest of the components in place. Then they could clear away the fat. This is a mouse brain before and after. The brain is now transparent to light, but it's also permeable to molecules, which means scientists can add molecular markers to highlight specific features. In this 1 millimeter block of hippocampus, excitatory neurons are green, inhibitory neurons are red, and cells called astrocytes are blue. The technique works in human brains too. This is a chunk of the frontal lobe of a 7-year-old boy who had autism. It's possible to trace the path of a single nerve projection through a forest of other cells. When the team looked closely in one layer of the cortex, they noticed ladder-like patterns where neurons had connected back to themselves and to other neurons. Similar abnormal structures have been seen in animals with autism-like behaviours. Being able to analyse brain structures like this and match them up with molecular information could help neuroscientists uncover how changes to the brain underlie disease. And it's not just the brain. The researchers say the technique could be applied to any organ.
Here you can see the results of the clarity technique. This 3D picture can show us the different neuron networks within the brain. This technique allows researchers to manipulate the different lipids within the brain in order to see neuron networks. And now, because of this technique, researchers can determine which type of cells are located in certain parts of the brain.